Good morning, Year 9. Hope you are all keeping well. For today's session, I've actually put together a recap on CB3a and B. So before we move on to the next lesson, I just thought it might be a good idea for me to go through what you should have already learnt from the presentations that have been posted. I'm actually going to teach it slightly differently to the order it was posted in the classroom. So you may recognize the image on the screen. Hopefully you would recognize that as DNA. It's got a very unique shape, very distinct shape. And we'll be talking about all of those things, or I will be talking about all of those things throughout this tutorial. So if we start off with DNA and essentially what it is and why it's important, I guess. It talks about the instructions for an organism. And it says these are found as a code in its DNA. So essentially what that means is how tall you're going to be, what color your hair is, what color your eyes are, the shape of your earlobes, etc. What proteins you're going to make, what proteins you cannot make, how high your metabolism is, etc. All of those instructions are actually found within the DNA, okay? And if we were talking about the DNA of an organism, and I mean the entire DNA of an organism, we call that its genome. So the genome is the entire DNA of an organism. In terms of the human genome, it says it's found on 46 long molecules of DNA, each in a chromosome. So DNA is wound up really tightly into these structures called chromosomes. Humans have 46 chromosomes. Your entire instructions, the entire code is held in those 46 chromosomes. They're divided into, or I say they're divided into pairs, but they're found in pairs. So actually we have 23 pairs of chromosomes, which makes that 46. The purpose, or the reason we have 23 pairs is because one set is maternal and one set is paternal. So essentially you've inherited one set from your mother and one set from your father, okay? And that obviously gives you your entire set of chromosomes. And we'll be looking at them in a bit more detail, probably in the next video tutorial when I start talking about alleles. So DNA, you should know what it stands for. You need to be able to spell this. It would normally be a 1-mark question, but it stands for deoxyribonucleic acid, okay? So do learn that. So that's what it stands for. Again, you can see that image there. We describe that shape as a double helix, if you don't know that already, okay? In terms of where the DNA is found, so again, this should be information you can bring or pull in from CB1. So DNA is found in the nucleus, and it's found in the form of these long strands called chromosomes, which we've obviously talked about already, okay? So it's found, so in the nucleus, all your genetic material is held in that. So in every single cell in your body where there is a nucleus, you're going to have genetic material in there, okay? And as we mentioned, 23 pairs in the human body. This is only for humans, 23 pairs. This number is not necessarily the same for other animals that you might come across. Again, plants also have chromosomes. Their numbers are going to vary from one species to another species. So 23 pairs is in the human body. So in all your body cells, there are 23 pairs of chromosomes, okay? And like I said, they take on that strand structure where the DNA's been wound up really tightly, forming, so those things are called chromosomes, as I've mentioned previously. I've already used the term double helix, so it has a double helix shape, and all that means is that it's two strands that are linked together. So you've got one strand here, one strand here. You obviously have your bases in the middle. They are connected, so a bit like a ladder. So if you take the ladder, and if you were able to twist the ladder, it would form a spiral shape. And that spiral shape is known as a double helix. It says the two strands are held together by bases. There are 4 bases. I think one of your do now tasks was to name the 4 bases. I think everybody got those correct. So just a reminder, we have adenine, cytosine, guanine, and thymine. So 4 bases, okay? They are held together, but there is a bond that occurs between the 4 bases. Again, that was a question in your do now task. Some of you were able to tell me what that bond was called. Some of you were not sure. Okay, so you were giving me answers that were not correct. So the name of the bond that holds those bases together is hydrogen. So it's a hydrogen bond, which I'm just gonna jump to there. A hydrogen bond that occurs between those bases, which holds it together. Another question I asked in the do now task was, how do they pair up? Again, some of the answers weren't very clear, so hopefully I'm able to make that clearer for you. So in terms of the way that they pair, so they have what we call complementary base pairing. It's really important to know that they always pair in the same way. So adenine always pairs with thymine, and cytosine always pairs with guanine. There are a number of ways you can remember it. Some people look at the letters. So for example, C and G are both curvy letters. So the curvy letters go together. A and T, more straight and pointy letters. That's one way. There are ways, you know, if you sort of look at the word, if I was to say apple tree, which is A and T, chocolate galaxy, and then the chocolate bar, so C and G, or you could say a car goes into a garage, C and G. So those are little ways that you can remember how they pair up, but they always pair up in the same way. A always pairs with T, and C always pairs with G, okay? So that is essentially the basic information that you need to know about the DNA, about the structure of DNA. In a couple of slides time, we'll go into the structure in a bit more detail. But before we do so, here is something on the screen where you've got one strand of the DNA and the bases are there. In an exam, a question could be to write the complementary bases for that sequence there. So again, you would obviously know that T always pairs with A. So your first complementary base would be A. And then obviously we've got A's there, so pairing with T, so it'd be ATT. We'd have GAC, GTCCA. Okay, so if we have a look at that, that's what your complementary strand would look like. Okay, again, like I said, in the exam, you may be given a very short strand, and you may be asked to provide the complementary strand for that particular sequence of bases. Okay, so as I mentioned, we're going to look at the structure of DNA in a bit more detail. So obviously very important here. We have, again, we've got the double helix structure here. So we've got one strand there, one strand here. Obviously you've got your bases, you've got them pairing, the complementary base pairing, and then you have bonding in between those bases. So again, you should recognize that structure there. However, what you might not recognize is just how it's been displayed. So normally when you see the strand of DNA, we normally draw this as a straight line, and we draw that as a straight line. Those are described as the backbone of the DNA, but actually chemically, that backbone is made up of a phosphate attached to a sugar, and that sugar is called ribose. So it's a pentose sugar, which means it's got 5 carbons, hence that shape. And specifically, it's called ribose, which you might recognize. Part of that anyway is in the word DNA. And that ribose sugar has a phosphate group attached to it, and it runs all the way down on both sides, and that is known as the backbone of the DNA. Again, we have the bases here paired correctly, C with G, A with T, again C with G. We've also got these lines here, they represent the hydrogen bonds. They're not physically there, you couldn't touch them, etc., but they are there in that they are holding those base pairs together. But what you will notice from this image is that there are 3 lines here and 2 lines here. And again, 3 lines here. That's because between guanine and cytosine, there are 3 hydrogen bonds. So 3 hydrogen bonds, between guanine and cytosine holding that base pair together. However, between adenine and thymine, there are 2 hydrogen bonds. Okay, so whether it was adenine with thymine or thymine with adenine, doesn't matter which way round the bases are written, they would only have 2 hydrogen bonds between them. Okay, so that's a more detailed version of the structure of DNA. Again, there was a 6-mark question in the presentation, which was asking you to explain the structure of DNA. That is a very possible question. I think this one has been taken from a previous exam paper. So all of the words that are in the keyword box there, you would be expected to use to describe or to explain, that structure of DNA. So it's more of a description of that structure. And then obviously talking about the bonding and the double helix, etc., that's been created. Okay, there's your model answer. You feel free to pause this and have a read, and then maybe in a couple of lessons time, you try and answer that question from memory in terms of all the aspects that you remember about DNA. So the second lesson that I've posted was the lesson, well actually, like I said, I've reversed the order, but the second session essentially would be on meiosis. So you've learnt about mitosis in CB2. So you know that it's a form of cell division. You know that it forms 2 genetically identical diploid cells, okay? And you should have had the opportunity to go through the meiosis presentation. So if you have, you should know that meiosis is also a form of cell division. However, it leads to the production of 4 cells. They are genetically different to each other. And actually, they're described as being haploid, which means they contain, I know it says here half the genetic information, which is correct. However, if we were going to write this in the exam, we were describing the difference, I would say that mitosis produces cells with 2 sets of chromosomes, but meiosis produces cells with 1 set of chromosomes. So if this was a human cell, then we would say mitosis produces cells with 46 chromosomes, but meiosis produces cells with 23. However, we've got terms diploid and haploid, which you are able to use. So again, scientifically, we want to use that terminology in an exam question. Again, a couple of other words here: gametes, scientific name for sex cells. The specific sex cells found in humans, the egg cell and the sperm cell. We've got the term zygote. So this is when the egg cell has become fertilized by the sperm cell. So literally, as soon as fertilization takes place, that single cell that's now produced is called a zygote. And the process of fertilization essentially is the egg cell and sperm cell fusing together. Okay, so again, you may recognize this image. This is just a quick overview of the process of meiosis. So we have our cell here, the nucleus, we've got the chromosome. So again, we know there's more than the number of chromosomes shown there. However, it would not be feasible to draw the 23 chromosomes because it would take up lots of space. So they've essentially got 2 sets of chromosomes here. Okay, so they've labeled it up as one set of chromosomes there and the other set of chromosomes, and obviously they've paired them up as well. So that would be one pair and that would be the second pair. So the process, the first few steps are very similar to that of mitosis. DNA is replicated, so the chromosomes go from being those single strands like this to be taking on the X shape that we know and recognize. They would obviously line up again in the middle and separate, so the pairs of chromosomes separate from each other. So as we can see, this orange and blue will separate from this red and I guess almost purple color, and they would form those 2 cells. But that's not the end of meiosis, so it's going to divide again. So in the second division, the actual X-shaped chromosomes are going to separate from each other. So essentially, if we just draw a line through the middle, cut them in half, we would end up with single strands that again we also know and recognize, and that's your 4 cells, and that number is haploid to the number we started with. So we started with essentially 4 chromosomes in this first image here, and then we ended with cells with 2 chromosomes. So this value here is haploid. This process takes place in the reproductive organs. So it happens in the ovaries, for example, in a female. These cells produced here, this 1, 2, 3, and 4, they would be essentially the gametes. So this would be 4 egg cells that would be used, could be used, in fertilization at some point. Okay, same process if we were producing sperm cells, so that's going to happen in the testes, so again the reproductive organs for the male, and these 4 cells here would be 4 sperm cells that are different from each other. Okay, and obviously millions of gametes are produced, so any one of those can be used in that process of fertilization. The next 3 slides just go through each stage, well, the 3 stages of meiosis in a little bit more detail. Again, you should have seen this, you should have made your notes, but I'll quickly run through it with you. So stage 1 is making, it says that the cell making the gametes has a full set of chromosomes. These chromosomes then copy themselves and stay attached to each other. So that's going from those single chromosomes here to these X-shaped chromosomes. So they're still called chromosomes, but they're just looking slightly differently in that this has replicated itself to look like that. Again, that one's replicated itself to look like that, and so on. Okay. So again, stage 1. Stage 2, the cell splits into 2 with each new cell getting half of the chromosomes. So this is where the pairs of chromosomes separate. So it's really important that we know it's the pairs of chromosomes that are separating. So that's your essentially, your first division. In the way that we've learnt mitosis in terms of prophase, metaphase, etc. In meiosis, we don't need to use those terms when we're describing these stages, but it is essentially the same process. So they line up in the middle, they get separated by spindle fibers pulling them apart. And then the final stage is the second division, where again the cell is going to sort of divide once more, and this is where the actual X-shaped chromosomes split into half. So one goes into one cell and the other goes into the second cell. And like I said, at the end of that process, we end up with 4 daughter cells that are haploid and that are genetically different to each other. Okay, it's very important that we use those terms if we were answering a question in the exam. And again, just reminding you what haploid and diploid means, if you don't know already. We've used it a few times now, but diploid means having a full set or 23 pairs of chromosomes, or you can also describe that as 2 sets of chromosomes. Examiners like you to be very specific, so they would probably be looking for it has 2 sets of chromosomes. And then haploid has that half a set. So in this case of the human, it would be 23 chromosomes, or like I said, 1 set of chromosomes. Okay, so that is the end of that presentation. So as I said at the beginning, it is just a recap on CB3A and B. I hope if there were any questions or anything that you were uncertain about, it is now being clarified, and you are happy that you have a good understanding of those 2 lessons. For your next lesson, which will be, if I remember correctly, on Wednesday, I will produce a presentation on the lesson about alleles in the same way and post that up for you to complete. Also, can I just ask, if I have not messaged you with your Easy Education assessment grade, please post me a private message on the classroom, and I can get that to you. For some reason, I'm unable to see who I've sent the grades to already. So really appreciate if you just drop me a message and say, and tell me that you haven't received it, and then I will send that to you. Okay. I hope you have a good day, and I guess you'll be seeing more of these presentations in the coming weeks.